In today's video, we're going to be covering the topic of gas exchange. So this is the second video in this series on gas exchange and gas exchange data. So our last video was on size and surface area, which is just a preliminary introduction into this topic. So if you haven't watched that one yet, I'd strongly recommend you going and having a watch because it will really help with this video coming up. So what we're going to cover in this video is just some basic information about gas exchange, and then we're going to look at how gas exchange occurs in 3 different organisms. So today we're going to look at fish, insects and plants, and in my next video I'll do humans because human gas exchange is quite a big topic. And it's quite complicated. You need to know it in quite a lot of detail. So to do all of it in one video, it will be very, very long. But let's get started and have a look at some of the common features of gas exchange. So gas exchange takes place over something called an exchange surface, and this could be something like a plant leaf or a fish gill. And gas exchange surfaces have several common adaptations. One of these is that they have a large surface area. The bigger the surface area, the more gas exchange can occur over that area and the more efficient the system is. They're also often very thin. If you think about a leaf, it's a very thin surface, and we'll have a look at the structure of a fish gill later, but again, they are very thin, and often these exchange surfaces are a single layer of epithelial cells. So next video, when we look at human gas exchange, you may have covered this in GCSE, but when you think about the alveoli in the lungs, they are a single layer of epithelial cells, and this provides a short diffusion pathway so that the gases that are being exchanged don't have to cross multiple layers of cells in order to get in and out of various parts of the organism. And if you think back to last video, I'm pretty sure we discussed Fick's law. If not, that's a good equation to look at, but basically for a system to be very efficient, it needs to have this short diffusion pathway. And then the third thing is that it maintains a steep concentration gradient across the exchange surface. Now what this means is, so a concentration gradient with gases like to move to an area of high concentration to low concentration. So if I had a can of deodorant, which is essentially a pressurized gas container, and I split that open, all the gas inside this highly pressurized container would essentially shoot out into the environment. So it will move from this area of very high pressure where there's a high concentration of molecules, and it will disperse into an area where the concentration is lower. So a steep concentration gradient is needed because if you think about in terms of oxygen, getting oxygen into a system, the steeper that concentration gradient is, so the higher the difference, if there's a much higher concentration of oxygen in outside of an organism compared to the inside of an organism, then when that organism breathes in or opens whatever cells are needed for gas exchange to occur, more oxygen will travel down that concentration gradient and into that organism. So in single-celled organisms, so things like bacteria, which are one single cell, gases are absorbed and released by diffusion through their outer surfaces. So bacteria need oxygen to grow and to replicate, and that gas or that oxygen will go directly through the bacterial cell wall into the inside of the cell, and any waste gases will travel out of the cell in the same way. So it just crosses from the outside of the organism to the inside or the inside to the outside. And these also have a large surface area, a short diffusion pathway, and a thin surface, so they don't need a specialised gas exchange system. But on the subject of gas exchange systems, let's have a look at our first example. So the first example we're going to look at is the counter-current system of gas exchange in fish. This, in my opinion, is the hardest one, which is why I've started with it. So on the right hand side of the screen, you can see that we've got a diagram showing the structure of a gill. So this gill is essentially this bony gill arch, which is supported by cartilage on the outside. And you can see almost these fan-shaped or wing-shaped structures sticking out of the edge. And these are called gill filaments. And they're stacked on top of each other to form this stacked filament structure. And these gill filaments essentially provide a larger surface area. And sticking up from the gill filaments, we've got gill plates, which also increase the surface area, but we'll go more into that in a second. So water has a much lower concentration of oxygen than air does. So for fish, they need to have special adaptations for this. So water, which contains oxygen, will enter through the fish's mouth and it will pass out through the gills. So essentially the fish will swallow water. It goes into something called the buccal cavity, which is the cavity just inside the mouth, and then the fish then closes its mouth, which creates a pressure gradient and essentially pushes the water over its gills. But the gills then have these special adaptations which allow it to extract oxygen from the water. So as we've said, each gill has these thin plates, these gill filaments, which we've already said about, which provide this large surface area for gas exchange. And sticking up from the gill filaments are these small plates called gill plates or lamellae, and this gives a further increase to the surface area. And these lamellae have many blood capillaries. They're essentially just a bundle of blood capillaries, and they have a very thin surface layer, and this speeds up diffusion. So as we've already said, blood and water flow in opposite directions over the lamellae, and this maintains a large concentration gradient between the water and the blood. I think I've made a typo here and I said oxygen concentration is always higher in the blood. It should be always higher in the water, because if oxygen concentration was higher in the blood, then that oxygen would then leave the blood and go into the water. So it should be oxygen concentration is higher in the water, and this increases diffusion of oxygen from water into the blood. So if we have a look at the diagram in the left-hand side of the screen, you can see these blue arrows, and the end of the arrow, the point, shows the direction the water's flowing in. So it flows from the mouth of the fish over the gills, and it goes out through this, the gills are covered by this little flap that opens and shuts called the operculum. So if you ever look at a fish and it's opening and closing its gills, that's the operculum opening and closing to release this, what is now water that's lost a lot of oxygen. So if you imagine the water, that's as it starts to flow over the gills, it's got 100% of the oxygen that it can contain, essentially, and it loses a percentage of that oxygen as it flows over the gill filaments and the gill plates, because oxygen is being lost from that water and going into the capillaries. So as you can see on the arrows, by the time it leaves, it's got about 15% of its initial oxygen. So that oxygen has been taken up by the capillaries and is now in the fish's circulatory system, so it can be transported to the fish's respiring tissues. And if you look on the right-hand side of my screen, I've got this this graph here, which is showing the comparative oxygen concentration of both water and blood as you go further along the gill plate. So at the start of the gill plate, which is going to be the furthest away from the operculum, this flap opening to release the water, the blood and the oxygen have their highest concentration, and the further you go along the gill plate, the lower the comparative oxygen concentration is, and that is because water is obviously losing its oxygen to the blood, but also because the blood on the or near the operculum, so sort of on this outer edge of the fish, if you like, is more oxygen poor blood. And you can see that the concentration of oxygen in water is always higher than the blood. So this means that the oxygen is essentially being transferred from the water into the blood. So moving on to our second example, we're now going to talk about gas exchange in insects. So insects have something called trachea for their gas exchange, and these trachea are essentially these little air-filled pipes. So if you have a look at my insect diagram in the bottom right-hand corner of the screen, we can see that we've got various bits labelled. So we've got these things called trachea and tracheoles and spiracles. So what happens is that the air enters these trachea through these pores on the surface of the insect, and these pores are called spiracles. So it enters into the spiracles and oxygen travels down the trachea, down a concentration gradient, and into the cells. So these trachea then branch into even thinner tubes called tracheoles, and then these tracheoles provide oxygen to the individual cells through their very thin and permeable walls. And we see a similar process in human gas exchange. So as air, if you breathe in air through your nose and mouth, it goes into a tube called the bronchi. You have two, you have a left bronchus and a right bronchus, and then these bronchi split into bronchioles, which are smaller tubes, so in a similar way to the trachea, splitting into the tracheoles. And then in humans, the bronchioles, the air diffuses directly into alveoli, which are these very thin, single-layered, spongy air sacs. But we'll go into that more in the next video, but I'm just highlighting that the processes are quite similar. So for insects, air goes into the spiracles, it travels down the trachea, these split into the tracheoles, and the oxygen goes directly into the respiring cells. And because it diffuses directly into these respiring cells, the insect's circulatory system has nothing to do with oxygen transport. It's a completely separate system. So in humans, obviously, we breathe in and deoxygenated blood comes into the lungs, it gets oxygenated, and that oxygen in the blood is transported to our cells. Does not happen the same way in insects. Its circulatory system has nothing to do with gas exchange. And CO2, which is the insect's waste gas, will move down its own concentration gradient, and this is then released through the spiracles. So now we're going to move on to our final example, which is gas exchange in plants. So on the right hand side of my screen here, you can see that I've got this, it's basically a diagram of a leaf, and the layers that exist within these leaves. So ignore the labels on the right hand side of the diagram because they are just pointing out the different organelles that exist in plant cells, and focus on the layers on the left hand side of the screen. So the way that leaves are made up, it's almost like a sandwich. So our outer layers on the top and the bottom of the leaf are this cuticle layer, and directly underneath this is the epidermal layer. So if we look at from the top of the cell to the bottom, we've got this cuticle with the upper epidermis just below it, and on the underside of the leaf we've got the cuticle with the lower epidermis just above it. And sandwiched in the middle is this mesophyll layer. And this is split into two. So we've got palisade cells, which are these nice sort of uniform square looking cells forming all lined up in a nice row. And then we have the spongy layer, which is much more diffuse cells. So just to confuse things and shake things up a bit, plants need carbon dioxide as their main gas because they need it for photosynthesis, and oxygen here is a waste product. But plants actually do need oxygen for respiration, and then CO2 is the waste gas. But the main goal of plants is photosynthesis, because photosynthesis produces the sugars that they need to survive and helps them to grow and then reproduce. So it's in the plant's interest to exchange as much CO2 as it can so it can carry out the necessary photosynthesis needed. So the main gas exchange surface is the mesophyll cell surface inside the leaf. And this provides a very large surface area, which you can see when you look at the layers comparatively. So even if we combine the cuticle and the epidermal layers, they are very small in comparison to the mesophyll layer. So it has this very large exchange surface. And the mesophyll cells, as we said, they're on the inside of the leaf and are sandwiched between the upper and the lower epidermal cells. And gases move in and out of cells through special epidermal pores called the stomata. On the singular, so stomata is the plural, stoma is the singular. So we go back and look at our diagram of our leaf layers, you can see in the lower cuticle there's this opening, and this is the stoma, and the stoma has two cells above it called guard cells. So essentially an open stoma is almost like an eye opening and shutting. So when it's open, you've got this small little sort of, if you think about your eyelids, when you open your eyelids, the layers separate and you get this sort of oval shape, which is your eye. And when they shut, obviously your eye is completely closed. And stomata work in a very similar way. So when they're open, they've got this sort of oval opening. But an open stomata also increases water loss. So they will open and close depending on how much water the plant is losing. So like we just said, open stoma looks like an open eye, this nice little oval shape where gas can flow through and be exchanged within this mesophyll layer. And when it's shut, it looks like when an eye is closed. It's just completely this nice little oval opening. It's completely shut. And this opening and closing of the stomata is controlled by cells called the guard cells. And the reason why stomata are only on the underside of the leaf is to minimize water loss. If they were on the upper side of the leaf, they have a lot more sun exposure, and this means that more water is going to evaporate. Now, they can be on the underside. It's beneficial for them to be on the underside of this leaf because it reduces evaporation, but this doesn't minimize how gas exchange can occur, because the gas can, in the air, gas is at the same concentration wherever it's surrounding. So whether it's on the top of the leaf or the underside of the leaf, the concentrations of carbon dioxide and oxygen will be the same whether it's on the upper side of the leaf or the lower side of the leaf. And that's why you only see stoma on the underside of leaves. So if we continue on and talk a little bit about how gas exchange systems are adapted to minimise water loss, we'll go a bit more into detail about plants, and we'll just touch on how insects are adapted as well to minimise water loss without the need to reduce gas exchange. This doesn't happen in fish because fish obviously live in a watery environment, and water is a very big part of how they get oxygen anyway, so we don't need to worry about water loss in fish because it won't happen unless they're plucked out of the sea. So in insects, they open and close their spiracles using muscles. So they'll use these muscles and they'll close the spiracles when too much water is being lost. And insects are also covered in this waxy waterproof cuticle in a similar way that leaves actually are covered in a waterproof cuticle. And they can sometimes be quite waxy. Insects are covered in this cuticle to prevent evaporation of water, and they also have these little hairs around the spiracles which can trap in moisture so that it's not lost in evaporation. And plants, as we've already said about the stomata, but stomata are open during the day to allow gas exchange, and this is because, as we've said, a principal thing that plants want to be doing is photosynthesis, and they need sunlight for photosynthesis to occur. So the stomata are open in the day for this gas exchange to occur and for plants to take in CO2 so that they can maximise their photosynthesis, but at night they then shut because there's no sunshine, there's no photosynthesis then taking place. So how these stomata open and close is that the guard cells, when water enters them, they become turgid, they become stiff, and they hold this stomata open. But when the plants become dehydrated, water will move out of those guard cells and they'll become flaccid, and this causes the stomata to close. And if you think about a plant, when you feel a well-hydrated plant versus a dehydrated plant, when they're hydrated, their leaves almost feel quite sort of not stiff, but they have this sort of rigidity to them. And they sort of stand up nicely and the plant looks healthy. When plants become very dehydrated, if you think about it, they start to droop, and this is because of these guard cells becoming quite flaccid, and they also have this almost rubbery-like texture. And we have these special plants called xerophytes, and these are adapted to warm, dry, and windy habitats, so something like a desert. So we look at the adaptations of xerophytic plants. Some of the adaptations they have is they have this stomata which is sunken into pits, and this helps to trap moist air. And it will reduce the concentration gradient of water between the air and the leaf, and reduces the amount of water diffusing out of the leaves and being evaporated. So essentially, if you think about a desert, very, very in the height of the day, it's often very, very hot. But as the air cools at night and possibly becomes a bit more moist, these stomata, when air travels over these pits that they're sunken into, they can trap this moist air in these sunken pits. And then in the heat of the day, when it gets very, very warm again, they reduce this concentration gradient between the air and the leaf, because if evaporation is going to occur, it's going to evaporate this moisture that's been trapped in this pit before it's evaporated out of the plant. As well as this, the epidermis has a layer of hairs, and this helps to trap the moisture around the stomata. So that's the point we've just covered, but air is trapped further because of hairs. The stomata are also often inside a curled leaf, and this protects from the wind. So wind increases the rate of evaporation. If you think about putting your clothes outside on a day that's got some sunshine versus a day that's got some sunshine and some wind, they're going to dry quicker because wind increases the rate of evaporation. Xerophytic plants also have a far reduced number of stomata, and this reduces the number of sites that are available for water loss. And they have this waterproof waxy cuticles that cover the stem and the leaves, and this helps to reduce evaporation. So to recap what we've covered, we've learnt that exchange surfaces are thin, they have a large surface area and a steep concentration gradient. We've learnt that fish have blood and water flowing in opposite directions, so this countercurrent flow, and this increases the diffusion of oxygen. Insects have spiracles, and this transports air directly into the insect's respiring cells. We know that plants require CO2 that shouldn't be respiration, that should be photosynthesis. And the mesophyll cell surface is used as the main site for gas exchange. We've also learned that plants exchange gas through pores called stomata, and these have guard cells which control the opening and closing of these stomata, and that when water is lost from plants, guard cells will lose water from the inside of the cell, they'll become flaccid, and the stomata will close. So now that we've covered this, let's have a go at some test questions. Number 1. Give 2 adaptations of gas exchange surfaces and describe how they benefit the organism. Number 2, how does the countercurrent system work in fish? Number 3, compare and contrast the processes of gas exchange in insects and plants. And what different adaptations do they each have that is beneficial for each organism? Number 4, what sort of climates do xerophytes grow in, and how are they adapted to these conditions? That's all for today. Thank you for listening.